Thank you for joining. In this episode of Outbreak, we'll take a look at coronavirus, how it infects cells, how it tricks our immune system. We'll take a look at the spike protein and mutations. There are 7 coronaviruses that are known to infect humans. 4 of them are very common and cause about 10 to 15% of the common cold. 3 newer ones are SARS, MERS, and the current coronavirus called SARS-CoV-2 that causes the disease COVID-19. These newer viruses have jumped from animal species to humans. It's unknown how many different coronavirus species exist in animals, but it must be in the thousands. So it is fair to assume that, in the future, we will likely see new types of coronaviruses jump from animals to humans, and will potentially see them cause other pandemics. Coronaviruses are called coronaviruses because of the spikes that stick out of the virus. In a cross-section, they appear to form a crown. This is a picture of a coronavirus attached to a cell and about to infect the cell. Next, we're going to take a look at how this works. To infect cells, coronaviruses need to enter the membrane of the cells. Enveloped viruses, like the coronavirus, achieve this by fusing with the membrane. What you are seeing here with the blue band of dots is a cell membrane with a couple of portal proteins and some receptors. Viruses will target a specific part of the membrane to attach to. In case of the COVID virus, it will attach to angiotensin converting enzyme 2, or ACE2. This is an important enzyme that sits on the surface of the cells in many organs, including the lungs. Its main function is to activate a process that lowers blood pressure. So if the COVID virus attaches in great numbers to ACE2, this process will become disturbed and people will get high blood pressure. Here we're seeing the COVID viruses floating around outside of the cells, and one of them comes close enough to the ACE2 receptor to bind with it. The spike being bound to the receptor will trigger a couple of shape changes in the spike to bring the membranes closer together to the point where they fuse and the genetic material of the virus enters the cell. I'm going to go over the spike in more detail, but before we do that, let's take a quick look at the genetic code of the virus and how it affects our cells. On the left, you can see the genetic code of the virus represented by letters. In reality, the code of the virus is a ribbon of nucleic acid with 4 different bases that correspond to the letters C, G, A, and U, or T if it were DNA. This code is read in groups of 3 to select specific amino acids to string together and build a large protein that way, such as the spike of the virus or other parts. On the right, you'll notice that the code is 29,883 letters long. For comparison, influenza is only half the size with about 15,000 bases, and HIV is even smaller with 10,000 bases. So 30,000 is not the norm for viruses, and coronaviruses are some of the largest enveloped viruses that we know of. These are the segments or genes of the virus. On the left in blue are some of the non-structural parts, and the red ones on the right are all structural parts. On the right, there's a gene called S that will construct the spike of the virus, E, which is an envelope protein, M, which is a membrane protein, and N, the nucleocapsid, which holds the genome of the virus together and protects it. When the viral genetic material enters a cell, it can be read directly by our ribosomes. Ribosomes are complex proteins that can translate the RNA code into proteins, not unlike a factory. They usually will construct all of the parts that our cells need, but they can also construct parts for the virus. A string of RNA will pass through the ribosomes and amino acids will be attached according to the code. The COVID virus will produce 30 viral proteins. While the names of the genes on the left look rather meaningless, for most of them we actually know what their function is. I will not go over all of them, but NSP1, or non-structural protein 1, will combine with our ribosomes and block them from reading code not related to virus production. So in this way, the virus can completely take over a cell and disable all communication with the outside world, like signaling our immune system that something is going wrong. In fact, when NSP1 is disabled, COVID becomes 100% non-lethal in mice. NSP8 and NSP9 interfere with protein transport to the cell surface, further limiting the cell's ability to communicate. And these together are supposedly part of the reason of cytokine storms. Interferon are a group of small signaling proteins that cells make in response to the presence of some viruses. Typically, a virus-infected cell will release interferon, causing nearby cells to heighten their antiviral defenses and get our immune system working. But this process is completely disturbed in COVID, leading to a delayed reaction that can flood the body with cytokines. These cytokine storms will cause inflammation and will ultimately lead to tissue damage. But this is not the only way that COVID tricks our immune system. Let's take a closer look at the spike protein now. So this is what the spike looks like on a molecular level. The bottom is attached to the virus and the top part will attach to a cell that has an ACE2 protein sticking out. The spike actually consists of 3 identical structures bundled together. When taking a closer look at the surface of the spike, you'll see these red ring-shaped molecules sticking out. Through glycolization, or covering itself with sugar molecules, the virus can hide from our immune systems. Our bodies will never attack sugar as it is our main source of energy. This is not a unique feature to SARS viruses. Influenza and other viruses are all covered with sugar molecules. But back to the single structure. There are a couple of different parts. The blue part is the N-terminal. Its function is not quite clear, but it is a location where antibodies can attach so that the virus becomes incapable of infecting cells. The red part is the receptor binding domain, or the location that matches up to connect to the ACE2 protein of our cells. The spike needs to change form to pull the membrane of the cells close to the virus. To do this, it will completely change shape, and when it has bound with ACE2, the top part of the spike will detach, and the teal-colored part of the stem of the spike will be cleaved by furin, while it is en route to the cell surface. This will make the central green part twist inside out, and a fusion peptide that is hidden in the middle of the structure will integrate with the cell membrane and pull the virus and cell together until their membranes fuse. But there's something particular about this spike. Our immune systems would easily be able to produce antibodies to block the receptor binding domain from attaching to ACE2, but this receptor is mostly hidden, and once in a while the virus will flip up part of the top structure to expose the binding site. And only one of these 3 structures will flip up at a time. Notice the green part flipping up here. By doing this, it can slow down recognition by our immune systems. But viruses also mutate. Fortunately, coronaviruses don't mutate very fast because of a proofreading mechanism encoded in its genome. The rate of mutation is about half that of influenza and about a quarter of the rate of the mutation of HIV. Still, with a high virus circulation, we're giving it a lot of opportunities to mutate. From the Wuhan strain on the left, we've now collectively produced a staggering number of mutations. Most of these mutations will not have much of an effect, but one particular mutation called N501Y has resulted in a virus that is much more infectious. This is an image of the top of the spike, and it compares the original Wuhan virus with the UK mutated strain. In orange at the center are the N501Y mutation differences. The changes are in the ACE2 receptor binding domain. This should normally be a well-conserved region, but this virus is not that well adapted to humans yet. These changes allow the virus to bind better with the ACE2 protein. In cyan, on the outside, are some deletions. These have an effect on how well antibodies bind to the spike and may make them lose efficiency. The effect of the blue changes closer to the center is still unclear. Other mutations may make the response of antibodies from a previous infection less performant and can increase the risk of reinfection. They may also cause antibody therapies to be less effective or even affect how well the current vaccines work ultimately. This is why it is very important that we limit the number of infections as much as possible. That's it for now. If you've liked this video, consider subscribing. We have more articles on COVID-19 and other diseases on our website. And while you're there, please consider donating to one of our projects. Until next time, bye.